All right, hi everybody. Thanks for showing up today, and thank you also for your patience with the quiz Canvas fiasco today. We're going to go ahead and get started with class now. And obviously I've had to postpone the quiz, so that is available till Sunday night now. It should be available pretty soon. And also your lecture notes for today are still due, just I extended that till Sunday night also. Okay, so I am recording today, so that's why I've got the video spotlighted, which I don't normally do. You obviously didn't take your post quiz. That will still be waiting for you. Module X is what we're working on today. So the first day of proteins, which is another really cool biomolecule. And I want to just remind you to check out MolView. And there's a short tutorial video. I'll show you some of that in class today, how the MolView works for proteins, but it's very different from the other molecules. And I think it also gives you a really good, not quite hands-on, but like eyes-on perspective on these big molecules. So definitely check out the proteins on MolView as you're studying. For Monday, you're doing Mod Y. And so that's the second day of chapter 16. You do have a notes upload, and you do have lab 14. There's no lab due this week, but lab 14 is up in case you want to work ahead this weekend. You've got Mod Y and Mod Z are both posted for you, okay? I think I already told you this on Monday, but I'm still way behind on grading, and I couldn't grade today. Actually, grading is broken right now on Canvas too, so I didn't have to grade this morning. I did other stuff. So yeah, that's that. All right. Any questions on the agenda here? All right, real quick, well, I have a poll for you in a second, but in the spirit of that it is Thanksgiving, almost tomorrow, you know I like Thanksgiving, I told you that before. It's a crazy one. It'll probably be the loneliest Thanksgiving maybe we've all ever experienced if you're sticking to your little house, but let's just throw into chat real quick something that you're looking forward to over the weekend, whether it's Thanksgiving specifically or otherwise. I love the food, and cooking has been really relaxing for me lately. So my thing into chat is going to be cooking and eating, but go ahead and just quick throw in whatever you're looking forward to this weekend, just so we have some reminders of what we have to be thankful for. Definitely looking forward to the break too. Parade. Simone, which ones do you watch? Do you watch the Macy's or something?
Yeah, that the Macy Day. Yeah.
Yeah. Are they doing it this year? Did they? We don't ever watch it.
I saw a commercial I think yesterday for it, so I think they're doing
Okay.
it.
Cool. I guess it's like you can do it safely because it's outdoors and easy to spread out. So cool. Ooh, new puppy, fun. There's like a couple of you there. All right. If you think of anything else you're looking forward to, throw it in there. Don't forget to take a little break. Breaks are good for re-energizing you, right? You come back smarter after a break, honestly, whether it's sleep or just some time away. So thank you all for sharing some of those fun things. Yeah, my brother-in-law keeps telling us we need to get a dog because that's what all the families are doing right now. And I'm like, no, I don't have like, I don't have time or energy or anything for one more small thing to take care of. So bless you all with your little puppies, but not me, not right now. All right, I also have a poll for you because I want to know what you're thinking and what you have questions on. So I'm going to go ahead and pop a poll up real quick. If you can just tell me where you have questions for today's stuff, then we'll make sure to hit on those today, okay? Oh yeah, in question 3, I asked you if you'd looked at anything on MolView yet, because I've had those suggestions up for the last couple classes. So I was just kind of curious if you had looked at them at all yet. Looks like a couple of you have. All right, so real quick here, get this flipped. Let me share your questions with you. Looks like we just have a couple questions. Did you watch the videos before class? Mostly, yes. Don't forget to upload those notes, easy points. And then maybe 3 to 5 of you have looked at those videos. Definitely take a look at them. I'll show you some more today to see those different molecules on MolView. So that is your feedback for me. Oh no. There we go. All right, so let me straighten this out again. So we are going to jump into chapter 16 then. And hopefully you took some notes on this first page. The first page is going to be our reference for some of the different colors. And yeah, just what we're going to see throughout this chapter. There's a fair amount of review in here. You'll notice the amides and peptides we saw in chapter 13 before. Acids and bases were also in chapter 13 and chapter 8. Ions, I've seen some of those. Side chains, I'll be using green here and there for the rest of the day. IMFs, we used those in chapter 4 and also in chapter 7. So we've got a lot of review built in to this protein chapter. I drew you this sketch of primary structure here where we have these amide or peptide bonds connecting amino acids. And I colored the side chains green. One thing I added yesterday, so you didn't get this in the video, was I realized I really should have circled these side chains up here in green also, so you can see how they relate to this primary structure here. So anywhere you see an S, those are side chains, and those can be circled in green. So they're all over the place, okay? And this just kind of looks like mayhem right now, but if I drew it, you should be able to draw it. This was our protein polypeptide that coiled up, and I end up with secondary structure holding the coils together, and then tertiary structure between side chains. And then if I have another polypeptide between a black polypeptide and a red one, I can have quaternary structure or connections between their side chains. So this is kind of getting ahead to where we're going to end today, but draw it, do it yourself kind of picture, because you tend to learn more from those sometimes when you actually draw it out yourself and see the connections. So questions or anything to clarify or be reminded of or anything on this page? Anybody? No questions? All right. Then let's go ahead and go to the next page. So slide 2. So slide 2 is, again, kind of a big picture of a couple different proteins. This is hemoglobin, and I'll show you this live on MolView in a second. Here's myoglobin. Here are some beta sheets that we'll see on the next slide, so you can see the difference between the helices, and then the beta sheets. And then over here we have our fibrous proteins. Fibrous proteins is what we have on our heads, if anyone has hair. And so the fibrous proteins are a good example of that. They're long and they're thin. Does your hair dissolve in water? No, you hope not, right? You don't want to get in the shower and have your hair dissolve. And so these fibrous proteins tend to be like protective and also are good for things like tendons, where you want them to just exist in a watery environment but not dissolve on you. So questions, anything to clarify on this page? I have a dedicated but questionless group of you here today. You all showed up, but you got nothing to wonder about. So anything at all you want to clarify or anything here?
I have a question, Professor, about the peptide bonds.
Okay, yeah, go ahead.
So can you, like, maybe this is actually a question more for slide 5. I'm not really sure. But so I know that they're amide bonds. Do you mind just talking a little bit about why those bond the rest of the protein together?
Yeah, so I don't know. Let me draw and then see if that helps you. So if I can put like actual atoms onto my protein, the backbone always has this pattern of NCC. So like these 3 little amino acids I can see here, I could actually start to draw out instead. And I'm always gonna see, so these would continue out like along a protein. I'm always gonna see an H here and an H here and then a carbonyl. So H, H, carbonyl, H, H, carbonyl, okay? And then down here on these middle carbons, this is where you're gonna see your side chains. So if I have maybe simple side chains, I'm just gonna make these all, I think it's alanine. So really, really simple. But this is kind of that same picture we had on the front page where here's my side chains. And then can you see the peptide bonds in here? I think that was you, Sarah, right? That was talking?
Yeah.
So can you see here is my peptide or my amide? And so this is the link between, if this is an amino acid here, here's the link from one amino acid to the next amino acid. So here's an amino acid, here's another one, and here's that peptide bond linking them. And if I keep going, I can see the same thing's going to happen again. There's that linkage. That was why we used that orange a bit ago. So here's another peptide bond. And here's the 3rd amino acid then with its own NCC and its backbone. So that's kind of a blow up of what I've got here. Does that get at what you were asking, Sarah, or do you want to add anything there?
Yeah, I guess my question was just like, what makes them a bond? Is it just because it's like because it is between those other parts of the atom, or why do we call it a peptide bond, I guess?
So we do, yeah, let me write that down. So we do call it a bond because it is like a physical connection. So it's definitely not an intermolecular force or anything like weak like that. It's physically like electrons shared with or electrons being shared there. In terms of like the etymology or the naming piece of it, it is just an amide bond if you're asking about a chemist's functional groups. I don't know where like the history of like peptide came from, but those are the exact same thing. A peptide bond or an amide bond is what we have boxed there in orange. So I don't know. Can you tell me anything else? I feel like I'm maybe not getting what it is.
No, that's okay. This is helpful. If I have another question, I'll ask. Thank you.
Okay. Anyone else have anything to clarify there?
Yeah, Professor. So when these bonds are occurring, it showed in the video that water was being released, H2O is being released. Is that the dehydration synthesis?
Yes, it is. We call it condensation in chemistry, but yeah, in physio or micro or whatever, they call it the dehydration synthesis.
Oh, cool. Okay.
Yes. And that
why aren't we seeing that H2O in the original bond then? If it's being released, is it just in there somewhere? Oh, OH.
So here's what you have. You have your amino acid 1 here, and then you have your second amino acid. This is the acid end, and this is the amine end. And when those two come together or condense or synthesize, then I pull out the water.
Okay.
Water leaves to kind of use my curved arrows. And then can you see how I'm going to smash these two together? So this and this end up becoming what I see up here.
Cool. Okay.
See that?
Yeah.
And so we did hydrolysis where we added the OH and the H back on, but now you can very easily see how you could also pull them out, and then where they both lost something, they say, hey, let's make a peptide bond so that we don't have a gap anymore. Does that make sense?
Yeah, that's cool, combining the what I learned from other classes and combining it to this and applying it. It's really cool. Okay. I was just making sure it's the same thing.
Yes, that was a really good connection. And it is, it's like, oh, now I like double get it from where I was before. What class do you learn that in? Is it physio or micro?
Physio, yeah, physio.
Physio, okay. Yeah, so great connection there. And so we would call this, let me just leave the word here. In chemistry, we call this a condensation reaction. And these I had mentioned them in chapter 13, or in physio, dehydration, because we're removing a water, right? And then synthesis, because we're building something big. That's what synthesis is to make something. So yeah, very good connection there, Brittany. Thank you. Anything else anyone wants to ask or comment on here? And so what I'm looking at, just to kind of get ahead of ourselves, is right now we're looking at the primary structure, right? Primary structure, we talk about being held together with these peptide bonds, where we're just like, okay, amino acid 1 connects to the second one, connects to the third one. And so that's what we're looking at, is the primary structure here. We haven't started coiling up into a helix or folding into a beta sheet. We're just like, what order do we have amino acids in? Anything else here you want to clarify? All right, good questions. Let me take this spotlight down, and I'm just gonna share my screen to show you a couple of these proteins, just the pictures on MolView. And I really strongly, again, wanna encourage you to check these out on your own this weekend also. So hang on while I adjust this. So this is one single amino acid, all right? This is alanine, and you can see how small it is, right? If I take this and I make it part of a longer chain, now I have insulin, okay? So the hormone that is gonna open channels up to let the glucose go into our cells. Can you see the alpha helix here? And I can also hold down with my mouse and I can kind of rotate it. So as I look on here, I can say here, if I had to circle and label, here's my alpha helix, right? The gray areas here, anyone remember what those are called?
Is it the beta?
No, those aren't beta. I'll show you beta in a minute. So we have alpha.
Is it the unstructured?
Yes, David, you're right. So this is what I call my unstructured region. I won't bother you by writing that out with my mouse, but unstructured means it's not coiled into a helix, or it's not flattened out into a sheet. So be able to circle and label on some of these structures as well. Questions there before we look at another one?
Yeah, where's the beta?
Okay, where's the beta? Hang on, let me click through because I have my tabs in order. We'll get to beta, okay? So here is a little bit bigger molecule. This is myoglobin. And you can see here lots of alpha helices, right? Still no beta sheets. We'll get to them, Brittany. But I do have this region here. This is, I think they're still called hemes and myoglobin, but I don't want to say that because I'm not 1000% sure. But this is what I would call the non-protein part of this molecule here. So I'm writing non-protein. It's C's and H's and O's and N's, but it's not amino acids, okay? So we have, in most of our proteins, we have the amino acid, the polypeptide, and then we have this other piece of it that actually kind of makes it work. A protein is kind of like a car, and then these non-protein regions are kind of like a driver. So the car is worthless without the driver there. You need this like active thing that actually lets you do some kind of action. So most of our proteins have some non-protein region, at least the ones that are like enzymes have that. So that's a myoglobin. And then another one here to show you, next one over, here's hemoglobin. Expand this just a little. And you can see it's a lot bigger, right? So myoglobin we saw on our sheet. Here's hemoglobin. This is something that you'll see in the video. I show you that there's some different ways that we can see this. So I can, here in MolView, and again, this is in the tutorial, but play around with it. I can color my secondary structures. So I can see there's a lot of purple here. That's the alpha helices, right? We keep seeing those coiled ribbons. You can also see the non-protein regions here, the hemes. There's 4 of them. Another thing I can do is if I want to see, well, how many strands are in here? I can click on chain is what I just did. How many colors do you see here of ribbons now?
4.
4. You got the green, the red, the yellow, and the blue. And if I unraveled those, I would have 4 separate subunits or 4 separate polypeptides. So that that chain here lets me see the different colors, one for each chain. If I go back to myoglobin and I do that same thing and say chain, there's only one color. So how many polypeptides does that tell me I have there? Yeah, just one. All right. One more thing I can do, let's go back to our, back to our hemoglobin. Oh no, I got to get to those beta sheets for you. This is a collagen. We'll just kind of skip through that. Here you go. Do you see something different there, Brittany? I'm picking on you because you were asking. So this is in that secondary structure kind of format. And so I can see some little bits of alpha helices, right? Anywhere I see purple, those are little coils. But then you can see these long, flat strand ribbon, the blue things. Those are your beta sheets. And so the color helps me see the difference between them. I still have some unstructured regions too, anywhere it's gray. Those are still unstructured, but can you see how the unstructured is very skinny? And then the beta sheets are a little flatter, or like wider and flatter. And that's because you've got like two strands of, or two lines of amino acids kind of flattening together and sheeting together. So questions, does that help you see the beta sheet there? Anybody? All right, let me go back to the hemoglobin to show you one more thing here. No worries. So if I go back to the hemoglobin here, you can also click on bonds. And did you see all those little lines show up? Can you see that on the picture now? So I have all these little, I don't know, looks like it grew little like spiky hairs or something like that all over. Those are the side chains. And so now I can suddenly see the side chains, those S's that we were coloring green. And so let me just circle a couple of them. Here's some side chains, here's some side chains, here's some side chains, okay? If I want to have tertiary structure, then my green side chains might attract other green side chains, and that would be tertiary structure, like you saw in the videos. If I want quaternary, now I'm gonna have to have one polypeptide attract another polypeptide, and so that would be something like my green side chains attracting my red side chains, and that would be quaternary. So between the two colors, between the two chains. Questions on that or anything you want to clarify or draw? All right. So the pictures are cool, right? They're very cool to look at. And I emailed you out. Let me see if I have it here. No, I think I pulled it down. Looking for cool pictures. I emailed you some cool pictures yesterday. You can see I've been like geeking out with lots of pictures here. All right, yeah, let's look at this one. So spike proteins are the protein that we've heard so much about lately. Here's the spike proteins on COVID, and I don't have more pictures here. There's more in the article that I sent you, but can you see some familiar structures in here now that we've been talking about the helices? So this is one of the spike proteins, and you can see the alpha helices in here. This is also out here, you can see my pointer, right? Can you see where I'm moving my little hand around to? So this is also other parts of the spike protein, and it's built, they're also built out of helixes and beta sheets. They're just not colored in here. So here they're talking about one of the ways that they made the spike proteins in the vaccines more stable was they added a couple of extra side chains in. I didn't know this, I was just learning about it this morning while Canvas was down. But they added some side chains in, and that made the spike proteins that are generated by the vaccines a little bit more stable. And so that's why they focused in on the middle here and showed the helixes in the middle, was to say, hey, here's some connections that we made. I think, let's see, the red. So where you see these reds here, these are prolines, which are some of your amino acids that you're gonna see. And from what I read a little bit further in, they tried a lot of different side chain additions, but the prolines seem to be the one that worked the best. So a couple of the vaccines, I forget, I think it's the Oxford one maybe, has the extra prolines in the spike proteins that are being generated by the vaccine to help it last, be a little more shelf stable. So like really cool applications of the stuff we're doing. And you know, I only know a little bit more than you guys about it, but even reading here, here's some of the terms you're gonna hear by the end today. Proline, salt bridges, those are those electrostatic attractions that you heard at the very end. Disulfide, remember those disulfide bridges? A cavity filling, that's different. That sounds like the dentist, but they filled in some gap here in the middle. But side chains are shown as red spheres, yellow sticks, red and blue. And if I can get out of that little gray thing, you can see like here would be a salt bridge. The reds are acid and the blues are bases, just like we've been doing, and showing one of those electrostatic attractions to help stabilize this. So it's kind of cool to have just enough vocab to read it and be like, wow, now I have a million questions. I need to learn more about this. But that's an everyday life application. And actually, Brittany, you were asking for beta sheets earlier. Here's another way that you sometimes see beta sheets. Can you see the little arrows there? So yeah. This is same, you know, it looks a little different from MolView. And here's another couple beta sheets, these long, flat kind of ribbons here. So it's not all alpha helices. There's some beta sheets tucked in there too. So pretty cool, right? Like cool stuff. And here's those side chains, right? So here's what we labeled S generically are there reaching across to hold these helixes together. Any questions?
So this is a quaternary structure, right? Because it's got four.
Yeah, the colors here are not actually separate strands, but in here I'll find on break. No, we don't have a break today, but I think there's one of the pictures I sent you, there's like 3 different chains. You can't see them with the color coding here, but the spike proteins are what they call trimers. So they have 3 different polypeptides. So you are looking at something that has quaternary structure. Yeah, because it's got the 3. So good question. Anyone else while we've got our application up? All right. So, kind of cool. Let's go back to the notes page then. All right, so that was this slide. The next one is here, right? Looking at our amino acids. And I only gave you some of these, I think, right? So here's our same favorite colors, our base, our acid, our side chain. And on this slide, I was telling you that you've got 4 different types of side chains to look at. And then once you can identify those, we can talk about changing the pH, okay? Let me find a paper here. So you also have, your next slide has all of the amino acids on it, right? So you have this sheet and I filled in a couple where I said, okay, let's look at the side chain and decide if it's non-polar or if it's polar basic. So I did a couple of those side chains for you on the next slide. That's something that we're not going to do right now, but that's something you should be able to do for homework, because in the quiz, you'll see, once we have access to Canvas again, on the quiz, you'll see that I'll give you all kinds of random side chains and ask you, are they polar, are they non-polar, are they basic, are they acidic, okay? So that's the first thing that we did on this slide, was be able to find the side chain and then classify it as one of these 4 types. So before we get to the pH, any questions on the side chains and what you're looking at for classifying them?
So Professor, whenever you have a NH plus, it's always polar basic, basically?
Yes, exactly. And be careful, it's always the NH plus in the side chain only, because these NH pluses are not in the side chain, and these are all side chain classifications. And that kind of comes back to this picture that we were drawing earlier. Do you notice once I build the polypeptide, there's no NH pluses in the backbone anymore because I've combined and those H's are gone. So the only thing that affects the polar acid, polar basic is whatever's left in the side chain here. Does that make sense? Good question. Thanks, Tiffany. Anyone else? Questions on our side chains, or if you worked 1 or 2 or weren't sure how to do them?
just to piggyback on what she was asking, so I was confused on the third one, whether it was polar basic or polar because of the H2N. I'm sorry, on the next page.
Oh,
These-
gotcha.
uh,
Here.
the third one, in the row, I was with the H2N. I'm not sure if that's just polar or if it's polar basic because it doesn't have a charge.
Yeah, great question. So Tiffany was pointing out to us, if I see that positive, I know that it's basic.
Right.
Brittany is following up to say, okay, now I see what looks like an amine, which I know is basic, but it doesn't have a charge on it. The fact that it doesn't have a charge is a really important hint. And you want to recognize this. It looks like an amine, but it has a carbonyl next to it. What functional group is that when you have what looks like an amine, but it actually has a carbonyl next to it? Anybody remember?
Oh my gosh. Is it a, it's not an, is it an amide? It's not an amide. What is it?
It is an amide or amide, however you want to say it. That was the orange color we used in chapter 13. This is an amide. And remember what an amide was, is it was an amine, but with that carbonyl next to it, or it was an N with a carbonyl next to it. And this no longer does not accept H+. Remember, this N is kind of tied into this carbonyl now, and so it doesn't want to accept an H+ anymore. And that's why it's not protonated. That's why there's no positive. So it won't accept H+. It doesn't have a positive charge. So I'm kind of thinking backwards here. And so this is not basic. And that was something we wrote down about the amides a couple weeks ago. And I said, just remember, these are not basic. This is why we wrote that down, because they're not basic, even though they kind of tempt you looking like it.
Polar.
Yes, so it would just be polar if I was classifying this, yeah. It can participate in a lot of interactions. It can hydrogen bond out to other side chains, but it's just going to be straight up polar. Does that answer your question, Brittany?
Yes.
Anyone else with side chain questions here?
Cleared it up for me.
All right.
The difference in positive and negative charges.
Sorry, can you say that again, Tiffany? It was kind of quiet.
That cleared it up for me. What you said, like if it's polar basic, it would have like a positive charge on the actual side chain.
Yes. And then I guess just to wrap up here, make sure you look for the opposite. Here's a negative charge, right? And so if I have a negative charge that shows up in my side chain, that's always going to be on a carboxylate is our word for that. Let's see. So it's like a carboxylic acid, but we put an 8 on the end of it when it's deprotonated. And so this is going to be our example of polar acidic, right? And we saw that on the previous page, but just to confirm it here. The acids are super easy because it also always has acid in their name. The bases are a little bit trickier because you've got the base and you've got the amides to keep things straight. So they're a little bit harder.
Would lysine be a polar basic because of that NH3 plus at the bottom?
Yeah, it would be. So here's my side chain. And then, yeah, you spot that positive. And so you're thinking, yep, this is going to be polar basic. Exactly. Anything else there? All right, so once you can identify those side chains, now you start to know what to look for in terms of changing the pH. Okay, so that was the thing we were doing on this slide here, was looking at changing the pH. So basically, we either make our solution really acidic. This would be like, for example, in your stomach, we have really acidic, a very acidic setting. So in the stomach, your pH is like about 1 to 2, depending on the moment. So that would be a chance to protonate your side chains or your backbone. And then over here, a base, you never want that base inside of you. You would die. This is like drinking drain cleaner or something like that. This is going to deprotonate things. But if we have an amino acid outside of the body, or maybe in a bacteria that lives somewhere really basic, then we want to be able to understand this. So let's work through one of these. One of these three, we can draw them out. Does anyone have a request for which one you want to work out at the different pHs?
The third one.
Third one, so 1, 2, 3, this third one?
Yeah.
Okay, it's so funny. That's what they requested yesterday too. It's like clockwork. Every class is the same.
Well, you did the first two for us. So.
Oh, I did this one too?
Mm-hmm.
Okay.
You did the pH one, but you didn't do the basic.
Okay, so I'll leave that one for you. We can work this one here. So at pH 1, if I look at what I've got going on here, can we plan ahead? Where do I have a spot that I can add a positive on? Anybody?
Like add a hydrogen?
Mm-hmm.
At the COO minus.
Yeah, so my acids, which is COO minus, can get protonated. That's going to be the pattern that I always see. Acids. This isn't really an acid anymore, but we're still calling it an acid. That is always going to be the one spot you can protonate. So you want to copy really carefully. Nothing else changes. NH3 plus CH, COOH. There's that H that I'm protonating. And then I just copy everything else. CH2, I'm just going to do a squiggle here, but I would copy everything else on down. So there's nothing to change except to protonate that one acid. Questions on that one at pH 1? All right, and then if I can slide over here and look at pH 12, so this was looking at taking 7 and moving it over to a pH of 1. For this next one, I want to again start at 7 and then convert it to 12. Okay, so looking here, are there any H pluses that I can pull off? Do you see any H pluses to pull off?
I'm sorry, Professor, what did you say? 7 and convert it to 12?
Yeah, so these are all at pH of 7, right?
Oh.
These are all 7. I didn't want to draw an arrow because this is my answer key, but my 7 turned to 1, and now I'm going to take 7 and jump over to 12. Okay, so I'm not going like through 1. That's what I'm saying. I'm saying it's two separate conversions. So what do you see here? Where can I deprotonate or pull protons off?
H2N.
H3N.
H3N, yes. And then anywhere else besides this?
H plus.
H plus. Yeah, anywhere I see those H pluses, those are the H's I can pull off. So if I slide this over and copy at pH 12, I'm going to have my bases get deprotonated, which means remove an H plus. And so if I copy, the carboxylate stays the same. CH, now I have NH2, and there's no charge anymore, right? Because I pulled the positive off. CH2, now I have to draw the whole thing. CH, NH, CH, double bond. And this is just an N now because I pulled that proton off of it.
All right.
That makes sense?
Yeah.
So it looks, everything with our biomolecules looks big and complicated, but it's actually kind of simple and then a lot of copying. So you find the places to pull the H's off and then you copy it well. Also with these, make sure you can put the words on them, right? So what am I working on? What functional group, and am I protonating or deprotonating? So those are kind of the two sides of it. Draw the picture, say the words. You want to be able to do both. Questions on that from anybody?
So you're only going to change the molecules that have a charge on them, regardless of whether there's a hydrogen, uncharged hydrogen, doesn't get changed.
Correct. I'm just looking for those charges. And so just to emphasize that with what I wrote here, I'm looking for where there were positives or where there were negatives. Those are my indicators. Hey, there's some place to add a proton or there's some place to pull a proton off because protons bring charges. So
Okay, that makes
yeah.
sense. Thank you.
Thanks for the questions. Anyone else with a question or anything to clarify here? All right, so good work on that. Let's go to our next page. So now we're on slides 5 and 6, kind of go together. On this page, you'll notice there's some ABCDs written. Don't worry about those yet. We'll talk about those in a minute. But this is a lot like what I did on slide 1. It's just kind of the book version of what we did on slide 1. And I found that because this was already printed, students wanted me to redraw it. So that's why I redrew it on slide 1. So hopefully having both of those will help you, okay? So we're looking here at the beginning of pictures that show us primary structure, secondary structure, tertiary structure, and quaternary structure, okay? And to keep our color coding going, you'll notice there's some green on here that was not here in the video. Anywhere I have these groups that are sticking off of my backbone ribbons, those are my side chains. So the same side chains that we saw on the first slide. So out in this space in between, these were the little hair-like looking things on the mall view pictures. These are all side chains. So you might want to add some green there to help you visualize those side chains, okay? Other questions on this slide? It's kind of going to go with the next one, so we'll also look at the next one and bounce back and forth between them. But any questions here? Or anything you want to clarify?
If nobody else has them, because I've been asking a lot, sorry.
You're doing great. Keep it up.
This disulfide bond right here,
Mhm.
is this only present in certain structures? Is this in every structure? This is the first time I'm seeing it in a protein chain. So I'm just wondering, like, when do those come in?
So they are in most proteins, but they're just like an option, like you can have them or you cannot. Anytime you have a cysteine, the cysteine, if you look back at your amino acid table, cysteine has an SH in it. So anytime I have two cysteines, they can actually reach across and form a bond. That was one of the ways they were trying to stabilize those spike proteins. The coronavirus ones I was showing you was they tried adding two cysteines because they're like, hey, this is the strongest tertiary structure connection is a covalent bond. So let's try that to stabilize it. It didn't end up being their favorite, but that's what we're looking at here is an actual covalent bond between two cysteines across a gap. So it is common. I don't know what else.
No, that's it. I was just wondering why it popped up now and just in this one, and if it was specific to this one, but it just is in them, right?
Yeah, it's just one of those things. It's one that gives all of these, right? Our hair is a protein. So the disulfide bridges, you can't break them. You can't flat iron those out or you can't curling iron those in because they're very strong, they're covalent. And so if you perm your hair with chemicals to make it straighter, to make it curly, then they use chemicals that will break those disulfide bonds and then put new chemicals in that will reform them in a whatever shape you want, straight or curly. And so those have to get like permanently or for whatever, a couple months, like altered by the hair chemicals. The rest of these, anywhere you see purple, if I decide I want straight hair today or curly hair, I can use heat to break those temporarily. And so you're not really breaking them, you're just taking apart those intermolecular forces. So a curling iron or a flat iron can undo that heat and then let it warm back up, go in the moisture, whatever, then your hair is just going to bounce back to the comfortable shape, whatever those intermolecular forces want. But the disulfide bridges, you can't break those with just a little bit of heat because they're a whole covalent bond. So, anyone else questions? Or Brittany, again, I don't mind if you ask lots of questions, but anyone else with questions here?
Well, there's more disulfide bonds in like curly hair then. Is that what you're saying?
The curls in your hair would partly be held in place by the disulfide bonds. My hair is pretty straight, so I could still have them, but they would hold my hair straight. And your hair is more curly, so you would have them, but they would hold a curly kind of orientation for your hair proteins. So it's not that, I don't think, I'd have to like triple check that. I don't think it's that one kind of hair has more or less. It's just mine choose to go straight and yours choose to hold curls. So yeah. Other questions on this page at all? So let's go ahead and connect this to page 6. And I've got more on my page 6 than you do, unless you kept going with videos. So all you need to know for your quiz next week is the first half of slide 6 here, okay? So the half that you can see. So don't panic that there's a whole other side to it. This is the words to go with the pictures. And that's, as you study, if you were here for student hours, I kind of took all the pictures from chapter 15 and added words to them. You might think about condensing all this to one page for yourself where you have the pictures and the words together, because that's what gets hard, right? Is when you sort words without pictures or you sort pictures without words. So make sure you can do both. Here's our levels of structure from simplest to most complex. We've kind of been talking about it all day, so just making sure you understand it. Primary structure is what order do I put these amino acids into? And then they start coiling up. So just to give you one more kind of visual, I have this like little red twine, I don't know, for Christmas. So this would be primary structure. It's just nice and straight, right? Secondary structure is when the backbone atoms start to be attracted to each other and I get a helix. Okay, so primary starts to coil up. That is secondary structure here. And it's gonna be these orange NHs and carbonyls that are attracted together to give me a helix. Now maybe next door, I also have another helix form, right? We saw all kinds of helixes in our pictures that we've seen so far. So that's secondary structure. And we circled some of those on the mall view pictures earlier. Now tertiary is gonna be same strand, right? It's all one strand, but when side chains from this helix are attracted to side chains from that helix, and it starts to pull them together. So that is my tertiary structure. And we'll see some examples back on the previous page. Does that make sense? You guys kind of see that connection there? Quaternary, I can't build quaternary if all I have is a red strand. So I would have to go grab another polypeptide that had maybe its own helices. And so now if I have two strands and they're able to connect, these attractions right here, those are your quaternary structure attractions, okay? Taking one strand to another, and they maybe hold together and move through your whole body together. So it's not like it's like a temporary thing. They're like happy to be in this hemoglobin, but it's quaternary structure when I have two polypeptides. So watch out for that one polypeptide is all of these, and two polypeptides is always going to be quaternary structure. Questions or anything to clarify here?
So that just confused me a little bit. So peptides are the amino acid chain. The polypeptides is more than one amino acid chain, and those polypeptides combine. Because I thought the quaternary was just 4 peptides.
No, it seems like you see the 4 there, right? Now my hand is all, hang on, let me just Google. I think the word peptide and polypeptide actually get used interchangeably, but I want to confirm that because I don't want to tell you wrong. Outside of chemistry terms, it gets a little bit confusing. So yeah. So technically, a polypeptide is our long chain of amino acids. That's what we're talking about here, is our long chain of amino acids. Polypeptides are usually technically 50 or more amino acids. And then a peptide, this is why you hear them used often interchangeably, is going to be between 2 to 50 amino acids. So these are actually both chains of amino acids. And then your amino acid is, I would say, your monomer.
Right.
Monomer, right? You just have one of those. So now I forgot what your question was, Brittany, in
Just
rambling
that, just that I thought I had it down until you just showed us those 2 polypeptides and said that these 2 are quaternary. Because it's 2 separate. I get that they're 2 separate ones, but I thought it was 4 together.
Gotcha. Yeah, so quaternary structure requires a minimum of 2 different strands, or you could have more than that. So it's just 2 or more polypeptides is how I would define those.
Okay.
Good clarification. Hemoglobin is a common example we all use, and that has 4 separate polypeptides, but they don't have to. Like the spike protein that I was showing you has 3, and that's still quaternary structure. So
I think it was just throwing me off with the vocabulary of primary, secondary being 2, tertiary being 3, quaternary being 4. Okay. So
yeah.
just get that connection. It's just the types. Okay. Got it.
Totally.
Okay.
Other questions or anything to clarify with the words here? So the last little connection I want to make for you here is I have letters here. You guys have these letters, I think, right? A, B, C, D, E, or A, B, C, D, no E. Do you have the letters here on these or no? Okay, so let's take each of these and just go back and forth to the previous page to make sure you can see where those are. Okay, so the disulfide bridge, which we labeled A here, if I can go back to this slide, you can see here it's called a disulfide bond. So bond and bridge are used interchangeably for those. And this is the one Brittany was asking about a couple minutes ago, the 2 sulfurs that are covalently bonded. This is the only type of attraction that's not just an attraction, it's actually a covalent bond. So it's the strongest of all of these. So everybody see that A, and you can label it A if you want, because that matches it to the next page. In order here on this page, these are in order from strongest to weakest. You'll see more about them in your next set of videos, next class. So these are strong because they're covalent. Electrostatic attractions, B is our next one. So if I can find one of those, let's see, electrostatic attraction is right here, okay? And this is where you have to be able to identify your side chains to help kind of think through these. On an electrostatic attraction, you're always going to have a positive and a negative. So that's why it's electro, like charges, static. I have charges. This is polar basic because I have the amine, right? And then here's polar acidic. And that's what you always build electrostatic attractions out of, is polar basic with polar acidic. So those charges we were looking at earlier, now they're important. Questions on that one or this disulfide?
The disulfide, that has to have the sulfur, right? If it doesn't have the sulfur, then it's not the disulfide bond.
Correct. You have to have both sulfurs actually. So it's a cysteine to another cysteine. It's always 2 sulfurs. Other questions? All right, so that was A and B. Let's go to the next one. C is polar attractions. So this is where you want to remember everything we learned in chapters 4 and 7 about polar attractions. Next class, we'll look at some different types of polar attractions, but you're just looking for especially O's and N's and H's, anything that's a polar bond. So here's an example of that, a hydrogen bond here. There's no charges, so it's not electrostatic, but I have O's and H's that can hydrogen bond with each other. That's a polar attraction. Here's another one. This is a weird one, so just ignore this. I won't really give you this again. This is backbone to side chain, and I am going to try and stay away from those. Not try, I am going to stay away from those. But here's a good example of side chains that are a polar attraction. Questions on that? All right, and then the last one, D here, is nonpolar attractions. Nonpolar to nonpolar is always dispersion forces. And so if I come over here, I can find a couple of those. Here's a bunch of C's and H's and other C's and H's. So that's dispersion forces, D. Or here's another one, all carbons and hydrogens, 2 side chains. And so those are also dispersion forces. Those are going to be the weakest, just like they were back when we learned in chapter 4 and chapter 7. Questions at all on those? All right, so then you can see the letters up here. Tertiary always has, or can easily have all 4 of those. You don't have to have all of them. Quaternary usually doesn't have the disulfide bridges. There's a couple exceptions, but in general, quaternary structure is just a little bit weaker, those attractions, so you don't have the covalent bonds for the most part. And so that is the end of what we have to look at today. Any other questions on anything here or anything else? All right, so if you look back to the beginning, let me just give you the quick 1-minute run through. Be able to draw your own pictures, because I did that and I had you draw along with me. So be able to draw and show those attractions. Your drawing doesn't have to be anything better than this, just not worse, please. And then we looked at some of those structures on MallView. So you'll see some of those types of structures show up in your practice quizzes, and I'll say, okay, is this an alpha helix? Is this the beta sheet? You need to be able to identify those. You also have a bunch of vocabulary here that you might have to sort, right? Sort the different types of proteins. Next page, we looked at sorting the side chains and also changing them at the various pHs. And you need to be able to do all the side chains on all the amino acids, okay? So that's part of your homework, is to go through all of those. Then we looked at, again, our big picture, be able to look at an attraction and identify what it is, and be able to not just look at a picture, but also do it in words, okay? Just don't worry about this side of the page. All right, so questions? All right, then that's it for our class officially. Thank you all for listening, showing up, asking questions. You'll have your practice quiz for this whenever Canvas gets up. They're still working on it. I just got an email from the IT person, but hopefully it should be soon. I've never had more than like an hour-long Canvas outage, so this is bizarre that it's been out all day. I'm gonna go ahead and stop the recording, and then I'll be here for student hours for half an hour if you have other questions, okay? Otherwise, have a really good Thanksgiving. Goodbye, everybody. And